Okay, the purpose of this recording is to explain some of the terminology around chromosomes and how the form of the chromosome is different depending on what phase the cell is in terms of the cell cycle. And so typically starting with G1, let's say, a chromosome is in, I'm going to make a drawing here, a form that we usually kind of draw as a DNA double helix. And so this is my drawing of a DNA double helix. So it's two complementary strands of DNA anti-parallel. And so we consider this one molecule of DNA. And sometimes we refer to this chromosome form as the monad form. It's a little bit of an unusual term, but I like to use it because I think it makes sense. In the middle of the chromosome, there's still a spot called the centromere. I'm going to darken it. It's actually a region of the DNA itself. And so that's one chromosome. So all of the chromosomes in the cell, if the cell is in G1, would be in the monad form. In S phase, the two strands of the monad separate, and you'll learn in your lesson on DNA replication, and each strand is a template to make a new strand. So here's the separated strands, and each strand becomes a template to build a new strand. So here's the new strand, and then here's the new strand. So at that point you have two DNA molecules, but these two are held together by one, and I'm drawing a ridiculously large centromere here. And so typically when you look at this chromosome under the microscope, you would refer to it as a dyad chromosome. So in S phase, the monad chromosomes become dyad chromosomes. And under the microscope, what you can usually see with a dyad chromosome is sort of almost a little X shape with these two thick DNA molecules, and then they're still connected by the centromere. But if you could really see them close up, you would see that each of these, what we now call chromatids, there are two chromatids, two sister chromatids that compose one chromosome, one dyad chromosome. So if a human cell is in G1 of the cell cycle, then it would contain, that cell would contain 46 chromosomes, but they would be all in the monad form. By the end of S phase, a human cell would still contain 46 chromosomes, but they would be chromosomes in the dyad form. In G2, the chromosomes stay in the dyad form. So I'm just going to write dyad form here. And then when the cell goes into M phase, the prophase is still a dyad form. Metaphase is still a dyad form. And metaphase is when they usually photograph chromosomes in a cell. And so that's why when you see a photograph of chromosomes, they usually they have that little X kind of shape, each of them, because they're in the dyad form. In anaphase, this is a transitional phase where the dyads are pulled apart, and each dyad is pulled apart into two monads. So you start off with dyads, but you'll remember that one spindle fiber attaches to each sister chromatid, and when those spindle fibers pull apart, they separate, each of the dyad chromosomes gets pulled apart into what would then be considered two monad chromosomes. So anaphase is an important point. S phase is when the monads become dyads, and anaphase is when the dyads go back to being monads. And by telophase, you have, if this was a human cell that was dividing, you would have 46 monads in the newly forming nucleus of this one daughter cell and 46 monads in the newly forming nucleus of this other daughter cell. And cytokinesis completes the split of the cytoplasm. And so by the end of the entire process, each of the daughter cells now contains 46 monads. So this cell would have 46 monads, and this cell would have 46 monads. And each of those cells then can go back into G1, which also has monads. So it's a continuing cycle. So I'm going to show you a website that I like. This is on the Learn Genetics at University of Utah. It's a very good site that you can get all kinds of tutorials on basic genetics, among other things. And I just wanted to show you this image here. This image is a karyotype, and it's a photograph of chromosomes from one cell. This would be a human cell. And then they've colorized, artificially colorized the chromosomes. But what I want to show you is when you photograph chromosomes, I mentioned before that the cells are typically in metaphase. It's just the easiest time to get a good photograph of the chromosomes. And so the chromosomes will be in the dyad form. And then they arrange the chromosomes in what we call homologous pairs. So here they've highlighted the homologous pair for chromosome 19. But what I want to point out is because the cell is in metaphase when the chromosomes are photographed, each of these little yellow blobs is actually a dyad. So this one on the right, even though you can't really make out the two sister chromatids because it's kind of fuzzy and the resolution isn't great, this is a dyad here on the left, and then this other yellowish blob would also be a dyad on the right. And so you would have, in metaphase, you would have two dyads representing the two copies of human chromosome 19. The same for human chromosome 20 and so on. So even though sometimes in the pictures you can't make out that this is, for chromosome 1, that this one on the left is actually a dyad form, it is, because the chromosomes are photographed when the cell is in metaphase. And metaphase cells always have dyad chromosomes. Cells in G1 always have monad chromosomes. So it's a rule in terms of what, depending on where the cell is in the cell cycle, that dictates whether the chromosomes would be found in the monad form or the dyad form. So this can be confusing, and I just wanted a chance to go over some of this terminology with you.